thank you to the organisers for this opportunity to tell you about Opthea's program to develop a novel biologic inhibitor for the treatment of wet age-related macular degeneration. So Opthea is a 100% owned subsidiary of Circadian Technologies, which is an ASX-listed publicly traded company in Australia. We're located in Melbourne, and our technology is really based on an extensive intellectual property platform in respect of members of the Vascular Endothelial Growth Factor family, or the VEGF family, namely VEGF-C, VEGF-D and VEGF receptor 3. Our lead compound is a trap molecule. It's a biologic compound that very potently blocks both VEGF-C and VEGF-D, and as a consequence is a potent inhibitor of angiogenesis as well as vascular leakage. OPT302 is in development for the treatment of wet AMD, but it does have a broad potential for development in other eye diseases as well, including diabetic macular edema on the basis of its mechanism of action, either as a monotherapy or in combination with the existing VEGF-A inhibitors that are currently approved for the treatment of this disease. We recently initiated our phase 1/2A clinical study. This is a study that we're conducting under an IND in US clinical sites, and we're actively recruiting wet AMD patients for that study right now. So the development strategy for OPT302 is to address the suboptimal responses associated with long-term single-agent therapy with VEGF-A inhibitors. Most importantly, the very large number of patients that experience a suboptimal visual outcome following anti-VEGF-A therapy, also persistent fluid on OCT and a chronic decline in vision despite receiving ongoing therapy. Now, clinical resistance to VEGF-A monotherapy may indeed be due to other members of the VEGF family, and VEGF-C and VEGF-D, which we target with our molecule, are key candidates for driving this mechanism of subresponsiveness. VEGF-C and D are both able to bind and activate VEGF receptor 2, and this is the key driver of angiogenesis as well as vascular permeability. It's also the receptor to which VEGF-A binds. And of course, VEGF-A is a target of Lucentis, Eylea and Avastin. Complete blockade of VEGF receptor 2 does not just require VEGF-A inhibition, but does also require VEGF-C and D. And we can achieve this through co-administration of a VEGF-A inhibitor as well as administration of OPT302. OPT302 can block VEGF-C and D, also binding to VEGF receptor 3, which in itself can also drive angiogenesis. And it stimulates angiogenesis through this pathway, which is VEGF-A independent. So our strategy here is to use it as a combination therapy with an anti-VEGF-A inhibitor to achieve a much more complete suppression of the VEGF pathway. This targets a mechanism of functional redundancy and mechanisms of subresponse to the existing inhibitors that are currently approved for the disease. So both VEGF-C and VEGF-D are critical regulators of angiogenesis. This is just a slide that demonstrates some of the data that's previously been published on the role of VEGF-C in modulating angiogenesis. You can see here that in VEGF-C deficient mice that there's a really a significant effect on the development of the retinal vasculature. In addition, VEGF-C is a very potent inducer of vascular permeability. Blood vessels that are chronically exposed to this molecule, VEGF-C, will go on to form endothelial fenestrations, which leads to fluid accumulation. And it follows, therefore, that if you block VEGF-A and VEGF-C, that you may therefore have a much more complete resolution of fluid at the back of the eye in AMD patients. The concept of subresponsiveness to anti-VEGF-A therapy being linked to upregulation of VEGF-C and D is now well established. There have been several reports in the literature, some of which have been generated by our own laboratories and collaborations, but also in independent laboratories around the world as well. And those reports clearly show that when you inhibit VEGF-A, both VEGF-C and D can be upregulated in a compensatory fashion in order to maintain signalling through that VEGF pathway. In addition, through our collaborations with Scapen's Eye Research Institute, we've shown that VEGF-C can be significantly upregulated and increased in the circulation of wet AMD patients compared to healthy controls and those patients with dry AMD. And we've also shown that VEGF-C levels at the back of the eye in wet AMD clinical specimens are significantly elevated and correlate with the severity of the wet AMD disease process. A little bit about the molecule. OPT302 is a soluble form of VEGF receptor 3. It's a trap molecule, and it comprises the first three extracellular domains of VEGF receptor 3 fused to the FC region of human IgG1. Therefore, it is very similar in structure to aflibercept or Eylea, but it does have a novel mechanism of action. It does not block VEGF-A, but it's very potent for VEGF-C and VEGF-D, preventing their binding and activation of both VEGF receptor 2 as well as VEGF receptor 3, as you can see there in the bioassay data at the bottom of the slide. We've also completed a series of pharmacokinetic studies with this molecule in both rabbits and cynomolgus monkeys, and we've shown that following intravitreal administration that this is a drug that has very good ocular pharmacokinetics, similar to that of aflibercept, with prolonged exposure in both the posterior and anterior segments. We've completed a comprehensive toxicology package of IND-enabling safety studies in both cynomolgus monkeys and rodents. This is to support our ongoing phase 1/2A clinical study where we're dosing patients on a monthly basis for 3 months. And we've shown in that toxicology package that this is a drug that's very well tolerated through intravenous bolus administration or through repeat dose intravitreal administration to monkeys as well as rodents, either in combination with Lucentis or as a monotherapy. In preclinical animal models of wet AMD, we've shown that this is a molecule that has very good ability, significant activity as a single agent to reduce the size of the wet AMD lesion in these particular mice. Consistently, we've shown data that makes it appears to be comparable in being able to resolve the fluid as well as the size of the CNV lesion comparably to Eylea or aflibercept, and used in combination, this is a drug that results in a further 15 to 20% added benefit in resolving wet AMD lesion size in this particular model of wet AMD. Moving on to our phase 1/2A clinical study, which is ongoing. This is a dose escalation study that evaluates the safety, PK and pharmacodynamics of OPT302 in combination with Lucentis in subjects with wet AMD. It's a trial that we're conducting in 2 parts. The first is a sequential dose escalation phase 1 open label study, and the second is a dose expansion phase 2A randomised study. In this clinical trial, we are actively recruiting patients with CNV, those that have either had previous treatment or are treatment naive. This is a little bit more detail on the design of our clinical trial. The phase 1 dose escalation component consists of 3 combination cohorts where patients receive 3 monthly injections of OPT302 at increasing doses in combination with monthly administration of Lucentis. At the top dose of the combination cohort, we anticipate opening a concurrent monotherapy cohort where OPT302 will be dosed on its own. The DLT window here period is of 1 month, but each patient receives 3 months of treatment with an additional follow-up at 6 months. The phase 2A study will recruit an additional 15 patients as a monotherapy and in the top dose combination. Standard primary objective of safety with a number of different clinical activity measures as secondary objectives. We anticipate primary data analysis from that phase 1 dose escalation late in the first quarter of 2016, and the primary data analysis of the phase 2 dose expansion in the second half of 2016 as well. And we anticipate recruiting approximately 50 patients in the phase 1/2A clinical trial, and it is actively recruiting. So in summary, this is a novel trap molecule. It's a protein biologic that has very potent activity against 2 alternative members of the VEGF pathway. Using it in combination with an existing VEGF-A inhibitor enables us to more completely suppress VEGF signalling, which we believe is a very key, important mechanism leading to sub-responsiveness to the existing therapies, and that by using this as a combination therapy, we can improve patient responses as well as reduce their treatment burden. Thanks very much for your attention.